Hi, my name is Matt Rettig. I'm a genitourinary medical oncologist with a specific interest in prostate cancer. My research interests extend from the lab to the clinic and back. And I have been at UCLA and the affiliated VA for over 20 years. So the question is, what is mismatch repair deficiency and how is it treated? So first of all, what is this entity? So the DNA in all of our cells in our body undergoes DNA damage all the time. It also undergoes spontaneous mutations, and that needs to be repaired because most of the DNA damage and the mutations that arise are harmful, deleterious to the cell. There's different types of DNA damage, generally categorized into single-strand and double-strand DNA damage. Most often, single-strand damage is the initial event, and there are different types of single-strand DNA damage, and one of them involves mismatch between bases, where instead of an A and a T, it could be A and a G. And there's a system of enzymes that are involved in mismatch repair. And when these enzymes are inactivated, typically genetically, occasionally epigenetically, there's a deficiency in the ability to repair mismatched bases and match them up again. And that's called mismatch repair deficiency. There are essentially 6 genes that govern the proteins that regulate mismatch repair. The most common ones that are mutated in human cancer are MLH1, MSH2, and MSH6. When these are mutated at birth in the germline, that is called the Lynch syndrome or hereditary non-polyposis colorectal cancer. And this can be associated with other cancers, including prostate cancer in men. In men with prostate cancer and metastatic disease, about 5% of the men have mismatch repair deficiency. Interestingly, for the most part, it's acquired. In other words, both alleles of the inactivated gene are inactivated somatically during life. A smaller proportion have a germline mutation where they were born with one of the 2 alleles of the affected gene inactivated, and the second one is inactivated during life in what will eventually become the tumor cell. So what happens when the cell has mismatch repair deficiency? That is, both alleles of the inactivated gene are dysfunctional. Well, you can't repair the mismatch bases, and mutations accumulate. And they accumulate so much that tumors that have mismatch repair deficiency have what's called the hypermutator phenotype. How can we leverage this phenotype for treatment? Now that we've defined it, what about treatment? So treatment has been evaluated across many, many tumors that have this mismatch repair deficiency. And the key is to understand that, first of all, the main therapy is immunotherapy with a checkpoint inhibitor. So why does that work? If you think about an immune response to a tumor, you think about it in 2 basic steps that are required. One is the immune cell, the adaptive immune response, namely T cells, must recognize the tumor as something that shouldn't be there. It's foreign. T cells are not geared to detect the normal host cells, but if they have a tumor cell that has lots of mutations, those mutations will result in altered proteins that have different structure that can be recognized by the T cell. And the second step is the killing step, where the T cell that has recognized the tumor cell actually induces death. So how do we take advantage of this? Well, in patients who have mismatch repair deficiency, the immune cell can recognize the tumor cell because it's mutated. It has abnormal proteins. There are neoantigens that are presented to the T cell. So there's an intact recognition step. The second step is the killing step, and that's where the tumor intervenes and says, okay, I'm not going to let you kill me. I'm going to express these checkpoints, the system whereby the tumor cell can put the brakes on the T cell response. And that most commonly leveraged checkpoint would be the PD-1, PD-L1 interaction. So in patients who have a hypermutator phenotype, when we use checkpoint inhibitors, in particular pembrolizumab, a PD-1 inhibitor, we see a very high response rate. And interestingly, for the very first time in the FDA's history, they approved pembrolizumab, a checkpoint inhibitor that targets PD-1, for mismatch repair deficiency, irrespective of the name of the tumor, irrespective of the kind of cancer the patient has. So whether it's colon cancer, prostate cancer, pancreas cancer, what have you, if the patient has a hypermutator phenotype, that patient is eligible for checkpoint inhibitor therapy. How do we detect the hypermutator phenotype or mismatch repair deficiency? So mismatch repair deficiency can be detected in one of a few ways. One is you can do somatic sequencing of the tumor, and you can identify the mutated genes. So if it's one of the mismatch repair genes, it's MLH1, you can see inactivation of both alleles, inactivating mutations of both alleles as an example. Sometimes the inactivation is not genetic, but epigenetic. So the gene sequence is normal, but there's epigenetic silencing, so the protein is not expressed. And that can be detected by immunohistochemistry to try to evaluate for the expression of the mismatch repair genes. There are two other important functional outputs of mismatch repair deficiency that can be detected. One is something called microsatellite instability. So throughout our genome, there are these short dinucleotide repeat regions, both within the reading frames and in non-reading frames, that have a given length. And in the context of mismatch repair deficiency, the lengths of those, what we call microsatellites, these short dinucleotide repeats, can change over time. And when they do, and they reach a certain threshold, then we call that mismatch repair deficiency. And it's defined by this, what we call microsatellite instability high tumors. The other way of defining mismatch repair deficiency, the final way, is just doing genetic sequencing through one of the commercially available vendors and identifying patients who have lots of mutations. So to be eligible for pembrolizumab, patients have to have 10 mutations or more per megabase, and that is sufficient to define that patient as eligible for pembrolizumab. By analogy, in prostate cancer, the median number of mutations per megabase in all comers with prostate cancer is only 2. But when patients have mismatch repair deficiency in prostate cancer, the number of mutations is typically much higher. So you can see there are different assays that can be used to detect mismatch repair deficiency, looking at the genes themselves in the sequencing, looking at the expression of the proteins by immunohistochemistry, looking at the microsatellite instability status, and also just looking at the overall mutational burden.